Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be doing an exam review for Biology 2421, Exam 2, which includes Chapter 5, Chapter 6, Chapters 15 through 18. Alright, so let's go ahead and get started. With Chapter 5, remember this chapter dealt with microbial growth and control. So I'm just going to flip through. And as always, please study all the material. Don't just go off of this review alone. But in this review, I will give you some hints as to some, of the more important things to focus on. But again, to do well, please know all the material, not just what I cover in this review, because this is just a review where I stress some of the more important points. All right, so let's go through. Obviously, if you see a term in bold, please know that the bold terms, all of these terms are important to understand. We spent some time talking about septum formation, so please know how a septum forms. So here you have the parts of binary fission. You should know cell elongation, septum formation. And remember that the chromosome of a prokaryote, there's a single circularized chromosome with a single origin of replication. And then you have DNA replication, DNA segregation, septum formation, and that results in two cells. Again, you have one single origin of replication on prokaryotic circular chromosome. Now you should know the steps to septation. Septation, of course, being the formation of cross walls between daughter cells. You should know these steps associated with septation. You should know about the FITs, or filamentous temperature-sensitive proteins, and each one's role during septum formation. So FITZ forms the FITZ ring around the center of the cell. ZIP-A anchors the FITZ ring to the cytoplasmic membrane. FITZ-A helps connect FITZ ring to the membrane and also recruits these other proteins, these other divisome proteins. All right. Oh, you should know about the Min proteins. Remember that the Min proteins localize the FITZ ring. Remember, FITZ proteins move back and forth, kind of like Knight Rider. Okay, the Min CDE proteins move back and forth. They oscillate back and forth, and because of oscillation of Min CDE, FITZ knows where to form. So you should know that Min CDE oscillate, and that directs FITZ location. You can see that process here. You should know about MREB. And it forms a spiral-shaped band around the inside of the cell. And you need to know why that's important. It's important because it localizes synthesis of new peptidoglycan. So new peptidoglycan forms around this spiral. Also, you should know that that's how the chromosomes segregate during binary fission. They move along this spiral. So MREB is involved in new peptidoglycan synthesis, and it's involved in chromosome segregation during binary fission. See, remember this was where the new peptidoglycan was formed, right at these dashed circles here. Crescentin, you should know that it's involved in the vibrio shape of bacteria. We talked about budding division. Please know what that means. Next, peptidoglycan biosynthesis. You should know the steps to peptidoglycan biosynthesis. You have autolysins that lyse the existing cell wall, the existing peptidoglycan. Then you have bactoprenol, which brings new subunits of, those NAG and NAM subunits to the cell wall. And then you have glycosylases, which add those new subunits. They make the glycosidic bonds between those subunits, introducing new subunits to the cell wall. So you can see here, let me show you what I'm talking about. Autolysins cleave the existing cell wall. Bactoprenol brings a new NAG-NAM subunit to the cell wall. And then glycosylases attach those new subunits by, dehydration reaction between these sugars to form new glycosidic bonds, right? And then you should know that once the sugars are included into the cell wall, you have to cross-link the peptide tails. And the cross-linking that happens between the short peptide tails of peptidoglycan, that's called transpeptidation. And you should know that penicillin, it inhibits this transpeptidation process. And that's how penicillin works to defeat bacteria. It does not allow bacteria to undergo transpeptidation, and that weakens the cell wall and the cells lyse due to osmotic stress. Please know these terms, the difference between sessile growth, planktonic growth. We talked about exponential growth. All right. This is something important to know for the exam. It's this concept of batch culture and microbial growth in batch culture. Don't forget that there is when you introduce bacteria to a batch culture, a closed system with fixed volume and fixed nutrients, first there's a lag phase. Let me show you. First there's a lag phase where there's little bacterial growth, followed by an exponential growth phase, followed by a stationary phase, and then followed by a death phase, or also known as senescence phase. So please know that these are the phases, the distinct phases of batch culture. Again, please know any words in bold here. These are just terms to know. I'm not going to spend a lot of time going over these simple terms. Here, this reiterates a point that we made in lab, and that is the viable plate counts. Two ways of counting microbial cell numbers. Viable plate count method. Remember this viable plate count method where you do a serial dilution of bacterium, and then you plate some known volume of each dilution, and then you find the countable plate. This is my countable plate to get my CFUs, or cells per ml of original stock. You should know, what's the main difference between a viable plate count like this versus turbidimetric counts. Remember turbidimetric counts where you're looking at the OD 600 or the OD 540 and measuring biomass based on turbidity? I need you to know, as we discussed in the lab, the difference between the two. So in viable plate counts, remember that you're only counting living cells, because only living cells will give you colonies. Whereas in the turbidimetric count like this, what you're seeing here, you're looking at total biomass. So this includes living and dead cells. All right, next we talked about temperatures that microorganisms grow in. You should know about the cardinal temperatures: minimum, optimum, maximum. You should know the different types of organisms based on the organism classification based on growth temperatures. So psychrophiles, you should know they grow best at 0 to 20 degrees C. Mesophiles, you should know they grow best at 20 to 45 degrees C, and so on and so forth, with thermophiles, hyperthermophiles. All right. So next we talked about pH. You should know that acidic pH means pH lower than 7. Alkaline pH is pH higher than 7. And pH 7 refers to neutral pH. You should know that acidophiles grow best in acidic pH. Neutrophiles grow best at neutral pH. And alkaliphiles grow best in alkaline pH. You should know about water availability. You should know that the higher water availability, so a water availability of 1 means pure water. And then a lower water availability means a solute-rich solution. And remember, bacteria usually prefer more water availability to grow, and they have a harder time growing with less water availability. And it's the osmotolerant bacterium that can grow over the range of water availability. You should know about osmotic concentrations of surrounding solutions. You should know that in hypotonic solutions, that means solutions of lower osmotic concentration. What does that mean? Like, for example, a hypotonic solution would be a solution of water, right? Pure water. A hypotonic solution is a solution of pure water, for instance. You should know that in this case, water would enter the cell, and unless the cell has a strong cell wall, the cell can swell to the point it bursts. So you should know that in hypotonic solutions, water enters the cell, and cells swell up and may burst. Whereas conversely, in hypertonic solutions, this means a higher osmotic concentration solution. So let's say I put my bacterium in high salt water, right? High salt water. Well, in this case, water will leave the cell through osmosis. Water will leave the cell, and this causes the membrane to shrink away from the cell wall. This is called plasmolysis. So please know that for the exam. You should know that it's the halophiles that are organisms that grow best at a little bit lower water availability, like seawater. Halotolerant organisms, you should know the definition of that. Extreme halophiles, please know the definition of that. Osmophiles, xerophiles. Great. Now we're talking about oxygen. Please know these terms: aerobes, anaerobes, facultative, aerotolerant, microaerophiles, anaerobes, aerotolerant anaerobes, obligate anaerobes. Please know the difference between these. Next, please understand the concept of reactive oxygen species. Remember this damaging, toxic byproducts of oxygen. Please know these. And then please know that organisms have adapted and evolved in order to grow in the presence of oxygen. Life did not adapt to grow in the absence of oxygen. Life adapted so that it could grow in oxygen. Does that make sense? It's a evolutionary adaptation to grow in the presence of oxygen. It is not a adaptation to grow in the absence of oxygen. And what's evidence of that? Remember, organisms that grow where there is oxygen and don't die to the reactive oxygen species of oxygen, those organisms possess special genes, such as the gene for catalase, peroxidase, superoxide dismutase, superoxide reductase. And you don't have to go in and memorize what all exactly these enzymes do, but you should know that these enzymes allow organisms to grow where there is oxygen. Please know these terms. These are very important terms for those of you who are going to go on to medicine. The difference between sterilization, inhibition, decontamination, disinfection. Remember, sterilization is the gold standard here, where you have killed or removed all viable organisms, including endospores, right? Very, very important sterilization. You should know that disinfection is not as stringent. You kill pathogens, but not necessarily everything. Okay, so know the difference between sterilization, which is the gold standard, and then disinfection. You should know about endospores and how they're resistant to a lot of different treatments. Please understand decimal reduction time. You know the concept of it. Please know the concept of decimal reduction time. Please know about the autoclave. What is pasteurization? All right, the autoclave. Next, we talked about radiation and filtration. Please know about types of radiation: microwaves, UV, X-rays, gamma rays, electrons. All right, ionizing radiation, we talked about that. And also, you remember something important I had mentioned, that you can use radiation to sterilize medical instruments or medical products like drugs, and also for food industry. Basically, if you have a product like drugs that you want to sterilize, but you know it's temperature sensitive, you don't want to put it in an autoclave and heat it up too much, you could actually bombard the drugs or whatever you're trying to sterilize for, you know, the medical purposes. You can bombard it with radiation, and that sterilizes without heating up and damaging the product. We talked about filtration. Please know the difference between cidal and static agents. Again, know the difference between sterilants, disinfectants, sanitizers, and antiseptics. You should know antiseptics. You're treating tissues. You're treating the living tissue. Sanitation is just probably the lowest standard, right? Because it's just reducing populations to levels deemed safe. Then chemotherapy. Please know that chemotherapy means killing or inhibiting growth of microorganisms within host tissues. And you should know that this chemotherapy includes antibiotics. If you take antibiotics, that's a form of chemotherapy in microbiology. Great, so that's awesome. We finished this chapter. Very interesting chapter. A lot of terms to know, but not too difficult. Let's hop to the next chapter. All right, let's carry on with chapter 6, covering microbial regulatory systems. Please know the difference between constitutive genes and regulated genes. Please know about the gene structure of a prokaryote. Remember that you have the structural gene, that means the part of the gene that codes for protein. You have the promoter, that's the region of DNA upstream of the gene. That means like in front of the gene. There's a stretch of DNA called the promoter. And the promoter is where the RNA polymerase attaches in order to initiate transcription. And don't forget that the promoter has two special sequences on it called the -10 and the -35 sequences. And those are recognition sites for the RNA polymerase to attach. And please know for the exam that, repressor proteins? Repressor proteins bind to the promoter, right? And that's how they repress transcription. They bind somewhere around the promoter or in front of the promoter at a place called the operator. Whereas activator proteins, remember activator proteins help turn on gene expression? Activator proteins bind upstream of the promoter. Repressor proteins bind to the promoter or just downstream of the promoter at a place called the operator. All right, and then when you do transcription, here's your mRNA. RBS stands for ribosome binding site, by the way. All right, so now please know the difference between negative regulation. So when you see negative regulation, you should know that it involves a repressor protein. When you see positive regulation, you should know that that involves an activator protein. You should know the definition of gene expression and gene regulation. And again, we talked about negative control. Here you're dealing with a repressor protein. Oh, and one thing I want you to know is this. With regard to negative control, usually you see negative control for amino acid and nucleotide precursor genes. Genes that are going to result in production of enzymes in the synthesis of amino acids for the cell or nucleotide precursors for the cell. All right, so here's your example of repression in action. This is an arginine, an amino acid arginine. This operon is being activated until the experimenter spikes in some arginine, and then the cell will switch off gene expression of those arginine synthesizing enzymes. Okay. You should know that induction means to turn on gene expression. So here with induction, usually you have induction with catabolic enzymes. That means enzymes that break stuff down, such as the lac operon, which synthesizes the enzyme beta-galactosidase, which breaks lactose down. Here's the example. Normally that lac operon is off until there is lactose around. Lactose is added, in which case you want to turn on the lac operon, so you're making the enzyme that breaks down the lactose, beta-galactosidase. Here is the example of the lac operon. Here is an example of a negative inducible operon. You should know this for sure, because the lac operon is a very important operon to understand in microbiology. You should know this, that the lac operon is an example of a negative inducible operon. Negative, remember, negative means you're dealing with a repressor. And then inducible means you can turn on gene expression. So inducible means you can turn on gene expression with the help of a inducer, a small molecule. So how does this system work? Let me go over this with you. Normally, there's a repressor protein, because this is negative control. There's a repressor protein bound to the operator, repressing gene expression of beta-galactosidase, LAC-Z, and the other lac operon genes. However, when there's allolactose around, this is a form of lactose, like a form of lactose that's synthesized by beta-galactosidase. The inducer allolactose binds to the repressor, and inactivates the repressor. This turns on or induces gene expression, right? So again, when you hear negative inducible, negative means you're dealing with a repressor. Inducible means you can turn on gene expression by adding an inducer. Does that make sense? Inducible means you can turn on gene expression, and gene expression is normally off. The opposite of that is, let me show you real quick and I'll come back, negative repressible. Okay, repressible means gene expression is normally on, but by adding a co-repressor, you can turn off gene expression. Did you follow that? Remember before I told you negative means you're dealing with a repressor, positive means you're dealing with an activator. But what does repressible mean? Repressible means the gene is normally on, but by adding a small molecule, by adding a co-repressor, you can turn gene expression off. So here you see the ARG operon, and gene expression is normally on. However, by adding a co-repressor, arginine, that binds to the repressor, activating the repressor, and now the repressor can do its job. Before, the repressor was not able to do its job. It was not activated. But when you added the co-repressor arginine, that activated the repressor to bind to the operator and block gene expression. All right, so I skipped a few slides. So here again is an example of negative inducible. This was, remember, like the lac operon example. The repressor is normally active. The repressor represses gene expression. However, if you add the inducer, such as allolactose, for instance, for the lac operon, that inactivates the repressor, allowing gene expression to commence. Please know the terms inducer. It's that small substance that induces enzyme synthesis or gene expression. The co-repressor, the substance that represses gene expression. And the effector is the term for both inducers and repressors. Co-repressors, that is. Okay, please know the term operon, obviously very important to know. Now we're hopping to positive control. Remember, positive control means we are dealing with a activator. And don't forget, activator binding sites are upstream of promoters. They are in front of promoters. So in the case of a positive inducible control system, gene expression system, positive means an activator is involved. Inducible means the gene is normally off. So the activator is normally not functional, it's not active until a molecule, an inducer, is added. The inducer binds to the activator. The activator can now bind to the activator binding site and help activate gene expression by recruiting RNA polymerase. Okay, and this is an example from your textbook of the MAL promoter. This is maltose enzymes for breakdown of maltose. And there you go. So again, positive meaning you have activator. Inducible meaning the gene is normally off. You need to add the inducer in order to turn on the gene expression. And here's a more generic cartoon. The activator is normally inactive. However, when you add the inducer binds to the activator, changing the conformation of the activator. The activator can then bind to the activator binding site, recruiting RNA polymerase, start gene expression. And you should know that the activator binding site can be upstream of the promoter, but it doesn't have to be right next to the promoter. It can actually be well upstream of the promoter as well, because the DNA could just loop back to help recruit RNA polymerase. Now, what's positive repressible? Remember, positive means you're dealing with an activator. Repressible means the gene is normally on. So if the gene is normally on, that means that the activator is always active. The activator goes to the activator binding site, recruiting RNA polymerase to undergo gene expression. However, when you add the co-repressor binds to the activator can no longer bind. So by adding the co-repressor, the activator can no longer bind, RNA polymerase won't bind, and you turned off gene expression by adding this co-repressor. You should know what the term regulon means. Okay. You may want to understand dioxic growth. Remember, you should know that bacteria utilize the better energy source first. And then once growth stops, they switch to the second best energy source. So you see, that's what dioxic growth patterns look like. They use up the better sugar first, so they're going to grow faster, utilizing that better sugar first, and then once exhausted, they can switch to the second sugar. Okay. I don't think I'm going to ask much about CAMP. It was interesting to discuss, but I don't need you to know how the mechanism of action of cyclic AMP. All right, we talked a little bit about two-component regulatory systems, a signal transduction in prokaryotes, where this involves a sensor kinase and a response regulator. Okay, the sensor kinase is usually a membrane protein, and a response regulator is in the cytoplasm. And remember the term kinase. Kinase means an enzyme that can add phosphate groups to some other protein. The kinases add phosphate groups. Okay, so here is an example of such a regulatory system. You have an environmental signal, some kind of hormone or ligand. This environmental signal binds to the sensor kinase in the plasma membrane. That autophosphorylates. This sensor kinase becomes phosphorylated, which means it self-phosphorylates. You got a phosphate group now attached to the cytoplasmic membrane protein, activating the cytoplasmic membrane protein, which then phosphorylates. This phosphorylates a response regulator. So now the response regulator is phosphorylated. And in this case, look what it can do. The phosphorylated form of the response regulator can bind to the operator, bind to the operator, blocking gene expression. So in this case, the response regulator is actually a repressor, but it's only an active repressor when phosphorylated by this two-component regulatory system pathway. So why are those important? Well, because there's almost 50 different two-component system pathways in E. coli that have been described. Examples include, phosphate assimilation occurs by two-component systems, nitrogen metabolism, osmotic pressure response, all kinds of different pathways. But the one we really touched on in class was the one for chemotaxis. Remember, how does the flagella rotate in response to chemotaxic signals? Do you remember that? It all had to do with Chi-Y protein. Chi-Y results in counterclockwise rotation and running of the bacteria. Remember, run and tumble. Chi-Y results in runs. Chi-Y, phosphorylated Chi-Y, results in tumbling. So remember how this worked? I don't need you to know how adaptation works. This is quite lengthy, but this is what I do want you to know for the exam. That when there's decreased attractant, so not as much attractant, you're trying to get to the attractant, but there's a decrease in attractant, then the MCP receptor, this receptor that senses attractant, triggers phosphorylation of the Chi-A, Chi-W complex. So now you've phosphorylated the Chi-A, Chi-W complex, which then in turn phosphorylates Chi-Y to phosphorylated Chi-Y. And remember, phosphorylated Chi-Y can meet up with the the flagella, the basal body of the flagella, and switching it to clockwise rotation for tumble, right? Because you want to tumble, you want to switch orientation. And once you switched orientation, then you can dephosphorylate Chi and run towards the food, run towards the chemoattractant, whatever the chemoattractant is. Remember, the chemoattractant could be light. In the case of phototaxis, it's oxygen in the form of aerotaxis. It's chemicals in the form of chemotaxis, etc. Then we talked about quorum sensing. Quorum sensing is a very interesting process. You should know that it is, when do bacteria undergo quorum sensing? You should know that quorum sensing is employed by bacteria anytime it makes more sense to do something as a group of bacteria than it does as a single bacteria, right? So bacteria do quorum sensing when it makes more sense to do something as a group. And the way they sense how many of themselves are around is by sensing this auto-inducer signaling molecule. Remember, the auto-inducing signal molecule acyl homoserine lactone, AHL. They release AHL until it's in sufficient concentration to switch on quorum-specific proteins, a quorum-specific gene expression. Now, what do I need you to know? There are these autoinducers. Remember AHL, there's also autoinducer 2, AI2, there's autoinducer 3. There's different types of autoinducers. And by the way, each species of bacteria has its own autoinducer molecule, right? If it can do quorum sensing at all, because not all bacterium can undergo quorum sensing. And what types of processes involve quorum sensing? You should know that the Lux operon, remember we talked about Alivibrio fishery? This guy is able to luminesce. It can glow with the Lux operon. So when there are sufficient of these Alivibrio around, they switch on the Lux operon and they bioluminesce. What else do you need to know? That here they are bioluminescing, in the pocket of the squid. You should know that virulence factors such as toxin genes can be turned on by quorum sensing. Okay, you should know toxins can be turned on by quorum sensing. You should know that biofilms are formed by quorum sensing. Okay, biofilms and plaque, right? Those types of things are turned on by quorum sensing. Well, very interesting chapter. All right, welcome back from break time with Gizmo and Wicket. Let's move on to let's carry on with our exam 2 review with chapter 15, the functional diversity of microorganisms. With our favorite, so here we are looking at Gizmo and different microorganisms and trying to understand how they are functionally different from one another. So we started with the cyanobacteria, and what you should know is that with the cyanobacteria, these are oxygenic phototrophs, so they get their energy from the sun, they produce oxygen. What else do we want to know? We want to know that they possess thylakoids, and they are responsible for a lot of photosynthesis on the planet. They contribute to 35% of total Earth photosynthesis, 80% of marine photosynthesis, and that's just these two classes, these two classes of cyanobacteria. Then we moved on to purple sulfur bacteria. And again, I know there's a lot listed here, but I try to cut to the chase and get to what's functionally different between these types of bacteria. So here, what's functionally interesting about the purple sulfur bacteria, I want you to know that they are anoxygenic, so they don't produce oxygen. They are anoxygenic phototrophs, and they use hydrogen sulfide as an electron donor for photosynthesis. So instead of using water as the electron donor for photosynthesis, these creatures use hydrogen sulfide as their electron donor for photosynthesis. So you could see why they don't produce oxygen. Because if you're not using water as the electron donor, you're not going to produce oxygen as a byproduct of photosynthesis. Then we talked about the purple non-sulfur bacteria. And let's see what I wanted you to know about the purple non-sulfur bacterium. Wide range of carbon sources. I just want you to know in this case that they are heterotrophs and they undergo photosynthesis. But there was nothing really too interesting here that I really wanted you to know. Let's just move on from these bacterium to the aerobic anoxygenic phototrophs. These ones are strict heterotrophs. And they are unable to fix CO2 and need organic carbon. So these are interesting because even though they do photosynthesis, they are unable to fix CO2 during the Calvin cycle. Instead, they use organic carbon. So that's kind of interesting. They are also anoxygenic under oxic conditions. So anoxygenic, unable to fix CO2, unable to undergo CO2 fixation during Calvin cycle. That's probably the most important thing about these aerobic anoxygenic phototrophs to know. Then we talked about the green sulfur bacteria. And what was interesting about the green sulfur bacteria? These ones oxidize hydrogen sulfide to sulfate. So they use hydrogen sulfide as an electron source. What about the green non-sulfur bacteria? The example here was chloroflexi. Other than that, there's really not much here that I need you to know. I just wanted to throw in an example of a green non-sulfur bacteria. Moving on to the sulfate reducers, sulfur reducers, sulfur oxidizers. Let's talk a little bit about these. So when we're talking about dissimilative sulfate reducers, these are interesting because they can couple oxidation of hydrogen or organics to reduction of sulfate. So they breathe sulfate. Does that make sense? They use sulfate as a terminal electron acceptor. So that would be an example of anaerobic cellular respiration. They breathe sulfate. That's what's interesting about the sulfate reducers. They reduce sulfate at the end of the electron transport chain. That means that they breathe sulfate instead of you and I, for instance, we would reduce oxygen. We would be oxygen reducers, if that makes any sense. What about the sulfur reducers? These reduce sulfur to hydrogen sulfide. So these breathe sulfur. What about the sulfur oxidizers? These are chemolithotrophs. They get their electrons from hydrogen sulfide. They get their electrons from these sulfur sources. So they get their electrons because they're lithotrophs. They get electrons from sulfur sources. All right, moving on to the nitrogen fixers. You should know nitrogen fixers, that diazotrophs are organisms that fix nitrogen into ammonia. All right, so I also would like you to know the difference between denitrifiers and nitrifiers. Denitrifiers grow by anaerobic respiration of inorganic nitrogen. So they use nitrate and nitrite. That's what they breathe. This is their terminal electron acceptor. So they breathe nitrate and nitrite. Instead of how you and I breathe oxygen, they breathe nitrate and nitrite. Nitrate and nitrite are their terminal electron acceptors. They use up nitrates and nitrites. And that's why they're called denitrifiers, because they end up removing these useful molecules from the soil, right? So they're constantly using up nitrates and nitrites, these important minerals in the soil, and then releasing gaseous nitrous oxide, N2O, and nitrogen gas. Nitrifiers, however, on the other hand, they grow chemolithotropically at the expense of reduced inorganic nitrogen compounds. So what they do is they oxidize ammonia, and they oxidize nitrite to nitrate and nitrate, and to nitrate and nitrite. They oxidize ammonia to nitrate, and they oxidize nitrate to nitrite. So what they do is they're nitrifiers because they produce nitrate and nitrite for the soil, whereas denitrifiers use up nitrate and nitrite and release it from the soil. Does that make sense? So denitrifiers, again, they breathe nitrate and nitrite. They end up reducing nitrate and nitrite to these other types of non-usable nitrogen. Nitrifiers do the opposite. They they grow chemolithotropically at the expense of reduced inorganic nitrogen compounds. So they take electrons from ammonia, they take electrons from nitrite, and they produce nitrate and nitrite and nitrate. Okay, so they provide nitrogen to the soil. Denitrifiers remove nitrogen from the soil. Oh, and just to make the point again, denitrifiers breathe nitrate and nitrite. They breathe it. That means they use it as a terminal electron acceptor. Nitrifiers get their electrons from ammonia and nitrite. Does that make sense? So that's how they grow, is by getting their electrons. They're lithotrophs. They get their electrons from ammonia and nitrite. I hope this makes sense for you. All right, moving on to other distinct functional groupings. We talked about the iron reducers. The iron reducers. This means that they breathe iron. They take electrons from some source, and then they get, at the end of the electron transport chain, they give iron the electrons. So they breathe iron. The iron oxidizers get their electrons from iron. They get their electrons from iron. Methanotrophs and methylotrophs. Methylotrophs grow using organic compounds lacking CC bonds. So we're talking they grow by using methane, for instance. Like they could get their electrons from methane. Methanotrophs, these use methane for growth. So the methanotrophs are a type of methylotroph. They use the methane for growth. They can oxidize methane. Predators. We talked about some predator species of bacterium. We talked specifically about Delovibrio. This was the one we focused in on. Delovibrio, which can be a predatory bacterium. It can insert itself into a larger bacterium. It can invade a bacterium, grow in length, and then divide by multiple fission and destroy the bacterium it preys upon. Then we talked about bioluminescence. Bioluminescence is emission of light, and there are some bacterium that can do this. All right, so that was a nice little functional rundown of different types of microorganisms. See how they're quite diverse in terms of where they get their energy, or what they breathe, or what they can do. That was a general sense of what's going on in chapter 15. Hopefully, you don't feel too inundated by information for chapter 15. It's not meant to overwhelm you. It's just supposed to give you some kind of awareness about the vast diversity of functional abilities of these microorganisms. So with that, let's move right on to chapter 16. All right, let's continue on with chapter 16, looking at diversity of bacteria. Here we're looking at different phyla of bacteria, and we're starting with the phylum Proteobacteria, and there are several classes of Proteobacteria: the alpha, beta, gamma, and delta and epsilon Proteobacteria. So what you need to know about the Proteobacteria is that they are all gram-negatives, and with alpha Proteobacteria, please know that these are oligotropic, which means they grow under low nutrient conditions. I gave you an example of an alpha Proteobacteria, Rickettsia. So Rickettsia is a causative agent of some human diseases, including typhus and Rocky Mountain spotted fever, and these are intracellular parasites. The beta Proteobacteria, I gave you an example to know about, and that's the beta Proteobacteria genus Neisseria. And the example I gave you was Neisseria gonorrhoeae, which is the causative agent of the sexually transmitted disease gonorrhea. Next, we moved on to the gamma Proteobacteria. And I wanted you to know that the enteric bacteria are these are gut bacteria, and these are examples of the gamma Proteobacteria. Remember, this is the phylum and class. This is the class of your gram unknown bacteria from lab, Enterobacteriaceae. I wanted you to know that there are mixed acid fermenters of enteric bacteria and 2,3-butanediol fermenters. And the main difference there was that the acids, acidic, lactic, succinic acid, are formed in large amounts in mixed acid fermentation, but in 2,3-butanediol fermentation, there's smaller amounts of acids formed. And then the examples I wanted you to know about of these enteric bacteria are Escherichia, like E. coli, for example, Salmonella, Shigella, Proteus, and Proteus is known for its swarming because it's peritrichous. And Enterobacter aerogenes, Klebsiella. I think the example I gave you, yes, is Klebsiella pneumoniae, which is one of the causative agents of pneumonia, one of several causative agents of pneumonia. We talked about Serratia as well. All right. Oh, the Pseudomonads are also an example of gamma Proteobacteria. Pseudomonas aeruginosa is an important species to know about. It's associated with opportunistic urinary and respiratory disease, especially in hospitals. The Vibrio-nales, Vibrio cholera is an important species to know about. It causes cholera in humans. And then moving on to the Delta Proteobacteria, the one I wanted you to know about here were the predators. the Myxococcales, and the Bdellovibrio. Epsilon proteobacteria. Here I wanted you to know about Campylobacter and Helicobacter. Campylobacter causes gastroenteritis and bloody diarrhea. Helicobacter pylori is the causative agent of peptic ulcers. Moving on to some gram-positives, the Firmicutes. So the Firmicutes lactobacillialis. This includes the genus Lactobacillus, which is common in dairy products. Then you have Streptococcus and other cocci, Lactococcus and Streptococcus. Streptococcus grow in chains of spherical bacteria, right? Lactococcus, these are dairy related to microorganisms. Enterococcus. Oh, next we talked about Streptococcus pyogenes, which is a causative agent of strep throat, and mutans, which causes dental caries. Remember, dental caries are the same as dental cavities. Listeria, these are also an example of the Firmicutes, Listeria, and they can grow well at low temperatures and can cause disease and foodborne illness. Staphylococcus is important. These are, remember, grape-like arrangement or cluster arrangement of spherical cells. The two I wanted you to know about here are Staphylococcus epidermidis and Staphylococcus aureus. These are resident skin microbiota. And then we're moving on to the endospore formers, right? Endospore former bacteria. The Bacillus and the Clostridium are the endospore genuses. I want you to know that Bacillus and Clostridium are the endospore formers. And I want you to know that not only is Bacillus, the genus Bacillus, known for formation of endospores, but it's also known for formation of antibodies, whereas Clostridium is only endospores, no antibodies. I meant antibiotics. So again, Bacillus is able to form endospores, and it is able to produce antibiotics. However, Clostridium is only able to form endospores without forming antibiotics. Next, we talked about mycoplasma. Mycoplasma, these lack a cell wall, so they do not have a particular cell shape. Mycoplasmas contain lipoglycans. Corynebacterium. The species I wanted you to know about these was Corynebacterium diphtheria, which is the causative agent of diphtheria. It's like a respiratory disease. The Mycobacterium, these are important to know about because they contain the mycolic acids on the surface. This is that waxy substance that makes a bacterium acid fast. So please know that the Mycobacterium contain mycolic acids, and that's what allows them to be acid fast. And here's an example species, Mycobacterium tuberculosis, the causative agent of tuberculosis. Then you have the actinomycetes. These are really neat. These are hyphal cells, which means filamentous cells, forming a mycelium, a grouping of hyphae form the fuzzy mycelium surface. And you should know that these are commonly found in soil. Then you have the streptomyces, and the streptomyces are also hyphae forming hyphal shaped cells, and they form spores. They can form spores. So here you can see streptomyces forming a spore. Here's the hyphal cell. The tip begins to curl, partition the tip, and then you form spores. Spores form at the tip of the hyphal Streptomyces cell. Streptomyces, again, is a soil microorganism responsible for earthy odor of the soil, especially after it rains. You know that earthy soil smell. That's the geosmin that's produced by Streptomyces. Streptomyces also produce a lot of antibiotics. So what I need you to know for the exam is that the Streptomyces and the Bacillus are the genuses that make antibiotics of prokaryotes. And it's the Bacillus and the Clostridium that form the endospores. All right, moving on to the Chlamydiae. So please note the life cycle of Chlamydiae. It's pretty interesting because there's two forms to the life cycle, two parts. An elementary body, which is a small, dense, cell resistant to drying and means of dispersal. And then the reticulate body, which is the vegetative form that divides by binary fission inside of a host cell. So you can see here's the life cycle of chlamydia. You have release of these elementary bodies, which are resistant. They then get into, they attack a host cell and get phagocytosed by the host cell. They then convert to reticulate bodies, which grow by binary fission, and then they form into elementary bodies, release, and the cycle continues. So this is the life cycle of chlamydia. And you should know that there are human pathogenic forms of chlamydia. These are chlamydia and chlamydophila, or chlamydophila. The chlamydophila stachyi, this affects birds and humans. Chlamydia trachomonas, this leads to blindness. This is a sexually transmitted disease which can lead to blindness, and human sexually transmitted disease as well. It's because there's two places where this affects. The bacterium can affect the eye, it can affect the privates. All right, that leads to the end of chapter 16. So a lot of material, but again, you don't need to focus on every little detail. Focus on the main characterizing features of these different microorganisms that I pointed out, and you should be good to go. All right, so let's move on to chapter 17. All right, with regard to chapter 17, it's a really short chapter because I kept it very simple, because we're looking at diversity of archaea, and as healthcare professionals, you're really not going to be dealing with archaea much, but I did want to give you just a small taste of what the diversity looks like for the archaea. One common, or the common traits of all archaea are ether instead of ester linked lipids, like you and I. Bacteria and eukarya have ester linked lipids. Archaea have ether linked lipids. They don't have peptidoglycan in their cell wall. They have other cell wall material besides peptidoglycan. Peptidoglycan is unique to the domain bacteria. You're not going to find peptidoglycan cell walls outside of the domain of bacteria. And they also are very structurally complex RNA polymerases, similar to those of eukarya. So remember, evolutionarily speaking, archaea, the domain archaea, is more closely related, has a more common ancestor with eukarya than it does with bacteria. Even though bacteria and archaea are both prokaryotes, you should know that bacteria is more distantly related to archaea, than they are to us, right? Archaea are more distantly related to bacteria than they are to us eukaryotes, because archaea and eukarya share a more common ancestor. So what are the hallmark traits of archaea? It's the only domain that can make methane, undergo methanogenesis. And many of these archaea live in extreme environments. You're not going to find them in, the human habitats much. The archaea are split into 5 major groups. These are the 5 major groups: Euryarchaeota, Crenarchaeota, Thamarchaeota, Corarchaeota, and Nanoarchaeota. And they are diverse. They inhabit many different environments. They can make methane again. They can inhabit high temperature environments, high salt, high acidity environments. Okay, and here is an idea of how they are related to one another. Okay, so that was about all I wanted to touch on. A highly diverse group, but mainly living in extreme environments, so not really organisms that you will encounter much as healthcare professionals. All right, let's move on to the final chapter of this exam 2, chapter 18. All right, let's start chapter 18, covering the eukaryotic microorganisms and the diversity seen within. Let's see. Remember the idea of the endosymbiotic hypothesis, or the endosymbiotic theory, that there's a lot of evidence showing that mitochondria and chloroplasts originated as bacteria long ago. And the evidence, you should know the evidence of this phenomenon, that mitochondria and chloroplasts contain DNA, and it's a circularized single chromosome, just like bacteria have. Eukaryotic nuclei contain genes derived from bacteria, showing that bacteria have been introduced into early eukaryotic cells. Mitochondria and chloroplasts contain their own ribosomes that undergo translation. Antibiotics that target bacterial ribosomes also impair mitochondrial protein production, which means that the mitochondrial ribosomes are similar to the ribosomes of bacteria. And then molecular phylogeny shows that in DNA of mitochondria and chloroplast resembles that of bacteria. So there are DNA sequences in mitochondria that are very similar to those sequences found in bacteria. So what these are evidence of the endosymbiotic theory, that the mitochondria and the chloroplasts originated as once upon a time bacterium. And then some of the protists underwent secondary endosymbiosis, where after engulfing a bacterium that became mitochondria or chloroplasts, that eukaryotic cell was then engulfed by another eukaryotic cell as a secondary endosymbiosis. And this gave rise to the green algae and the red algae. So you can see here, this was primary endosymbiosis, where the mitochondria and chloroplasts came from. So you have cells with mitochondria and chloroplasts. And then the red algae were engulfed during a secondary endosymbiotic event to give rise to the dinoflagellates, the apicomplexans, and the staminophiles, whereas the green algae were engulfed during a secondary endosymbiotic event to give you the euglenids and the chloroarachniophytes. Tongue twisters. Now, there are some groups of eukarya to know about, some supergroups of eukarya. Let's talk about the supergroups here. These are the different groups of protists you should know about: excavata, alveolata, staminophiles, resiaria, and amoebozoa. Let's talk about these. Excavata. The excavata, what you need to know about the excavata are that they are diplomonads, which means they have 2 nuclei of equal size. Okay, and then I think I gave you an example here: Giardia intestinalis, which causes giardiasis, which is a waterborne disease. parabasalids, which they contain a parabasal body. Those were the ones I gave you as examples there. So you can see here the Giardia intestinalis with their diplomonads. You can see the 2 dots here represent their 2 equally sized nuclei. And then here you have the parabasalid, and that was another example. And then we looked at the kinetoplastids, and the examples I wanted you to know here were Trypanosoma brucei, which causes African sleeping sickness. You should know about this Trypanosoma causes African sleeping sickness. Euglenids, these are non-pathogenic. I don't know. Oh, euglena. Yeah, euglena is a great example of euglenid that you should know about. It is a phototrophic protist, so it undergoes photosynthesis. Then the alveolata. The alveolata are characterized by the presence of alveoli, which are sacs underneath the cytoplasmic membrane. Here's an example of alveolata ciliates, such as the paramecium. You should know that paramecium is a ciliated protist. Okay, I don't think I want to touch too much on Balantidium coli. Next, moving on to the dinoflagellates, and again an example of alveolata. Here, the dinoflagellates have 2 flagella with different lengths and insertion points on the cell. Here I have an example of a dinoflagellate, though you don't need to remember its name. What you do need to know is that dinoflagellates are responsible for red tides. They're also responsible for the toxic spores that kill off fish and can cause food poisoning. And also you can see bioluminescence with the dinoflagellates as well. They can bioluminesce during blooms at night as well. You can see the blue lights. Apicomplexans, these are obligate parasites of animals. So here's an example of a apicomplexan, Plasmodium, which causes malaria in humans. Next, talking about the straminopiles. These include the oomyces, diatoms, golden algae, brown algae. Diatoms. You should know about diatoms because they are very important for production of oxygen on the planet, for photosynthesis. And they look very interesting because of their frustules made of glass. They have these glass frustules. The oomyces, what do we want to know about them? They're called water molts, and they grow with filamentous growth. Here's the golden algae. The golden algae and the brown algae are photosynthetic, and they have their golden brown pigmentation because of a pigment called fucoxanthin. All right, next we have the Rhizaria. These are distinguished from other protists by having thread-like pseudopodia that they use to move and feed. I don't need you to know all the different examples here. I threw them in so that you get an appreciation for what their tests look like. They have these test structures, and they have radial symmetry. So, but you don't have to worry about that. Let's move on to amoebozoa. This is a little bit more interesting. So the amoebozoa, you got the gym amoeba, and you have amoeba proteus, which is your standard textbook amoeba. And what you need to know about the amoeba is that they move by pseudopod motility. That means they can extend a false foot, which is part of their plasma membrane, and they use that for motility. There's entamoebas like slime molds. And what do we need to know about slime molds? They're interesting because they have similar life cycle to molds, to fungi. So this is what slime molds look like on a petri dish. All right, lastly, we talked about fungi. So let's go through about fungi. Fungi grow by hyphal arrangement. Their cells are hyphal, so filamentous. They're heterotrophs. In fact, they are saprophytes, which means they break down dead organic matter. They have conidia, which are those spore-forming structures, the asexual spore or conidia. Then the hyphae form mycelia. This is the fuzzy surface of the fungi. The mycelia is the fuzzy surface of hyphae. They can form mycorrhizae in conjunction with plant roots to help the plant root obtain certain nutrients like phosphorus and nitrogen. Okay, here's fungal cell growth. You have hyphae. The hyphae form a stalk. At the end of the stalk, they form a conidiophore, and then the conidia spores release, germinate, and form hyphal cells. Here's Aspergillus. Remember Aspergillus with its palm-like conidia spores? Okay. Oh yeah, again, you should know that fungi are saprophytes. Fungi are saprophytes. They can break down dead organic matter. And then some fungi form fruiting bodies, like the mushrooms. The part of the mushroom you see is the fruiting body. Fungi can cause diseases in plants and animals. These are called mycoses. Mycoses are diseases caused by fungi. Okay. What else did we talk about? Oh yes. How do fungi reproduce? They can grow and spread with the filaments. They can grow with their hyphal filaments. They can then undergo asexual production of spores with the conidiospores. And then they can also bud. They can bud as well. These are three asexual means by which they can divide. But then they can also form sexual spores as well, like the zygospore, which we learned about in lab, ascospores, basidiospores as well. So here you can see baker's yeast, Saccharomyces cerevisiae, undergoing a budding. Okay. The earliest fungal lineage is the chytridiomycetes, known as the chytrids. So here, let's talk about the chytrids. The chytrids are the earliest of the fungi. Okay, the chytrids are the earliest of the fungi, usually found in moist soil and fresh water. Okay, so know that. Then we have zygomycota. The zygomycota, I want you to know that they include Rhizopus stolonifer, which is the black bread mold. And we learned a lot about Rhizopus stolonifer in the lab, remember, with its different kinds of asexual spore. The sporangiospore was the asexual spore, and the zygospore was the sexual spore. Here you can see zygomycota, the Rhizopus. Moving on to glomerulomycota. Glomerulomycota, all known species, form endomycorrhizae with plant roots. So these form a symbiotic relationship with plant roots. They provide certain nutrients to the plant root. The plant root returns the favor with sugars. Then we have the ascomycota. These are the sac fungi, and they include Saccharomyces cerevisiae, the baker's yeast. Here you can see the Saccharomyces cerevisiae, the baker's yeast budding. Remember, it grows by budding, budding asexual reproduction, and then they divide by budding. Now, here's really interesting thing about Saccharomyces cerevisiae. Not only do they grow by budding, but this is interesting to know on the exam. that yeast can grow by budding either as diploid cells or as haploid cells. They bud either as haploid cells or as diploid cells, but they can then, if they're diploid, they can undergo meiosis to form haploid cells, and if they're haploid cells, they can undergo cell fusion to form a zygote, to form a diploid cell. Okay, then we lastly talked about the Basidiomycota. The Basidiomycota include the mushrooms, toadstools, rusts, puffballs, etc. And these are, you have the fruiting body that you can see above the ground. Oh, and that was the last of the fungi, but we have one last group of microorganism to talk about that's eukaryotic, and these are the Archaeplastida, including the red algae and the green algae. Let's touch on these. The red algae. The red algae are also called rhodophytes, and what you need to know about these is that they get their red color due to phycoerythrin, which is a accessory pigment molecule. And lastly, we talked about the green algae. These are called the chlorophytes. These undergo cellular photosynthesis, okay, and they produce oxygen. They are closely related to plants. And there's a huge diversity of these green algae. All right, and that's it for this chapter. Again, a lot of material here, but focus on those important points. Sometimes I throw material on here because it's interesting or because it adds to the story, but you really don't need to know every little thing. Know at least anything that's in bold. Know anything that I highlighted in this review. But don't worry that every little detail is going to be on the exam, because this is quite a bit of detail. And again, a lot of it was there simply to give you an appreciation for the diversity of the microorganisms we're talking about here, because life is quite diverse. So with that, hopefully this was helpful to you. Please study hard and prepare the best you can, and I wish you luck on your exam.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.